Okay, very good evening to all the participants and respected resource person, Dr. G.BHAGEERATH sir, dynamic personality, expert in molecular biology, especially plant genetics. Sir has working in uh, GDC, Government Degree College, Rangasaipet, Warangal district, as a assistant professor, head department of botany. Since 2021, Sir has given his valuable services to our young aspirants, those who are participating in CPGET entrance examination. They are get benefited from Sir's classes. So that's why we would like to invite Dr. G.BHAGEERATH sir. Please welcome sir and share your screen. Start your session. Please welcome sir.
Thank you, sir. I welcome to this CPGET online entrance coaching. In the continuation of yesterday's class, today we dealt with chromosomal mutations. In the last class, we discussed about point mutations or gene mutations, as well as little information about cytoplasmic mutations also. So today in this class, we can explain about the chromosomal mutations. Every biology student know very well the typical chromosome structure consisting chromonemata strands, primary centromere, secondary centromere, as well as SAT region and telomeres. So when these chromosomes undergo different changes, when some type of factors influenced, so these changes are visible. So the visible changes in the chromosome structure, it involve in two type of alterations. One is changes in gene number, another one is changes in gene arrangement. So in these two aspects, the chromosome is undergo alteration or go for the changes. So such type of changes we can call as structural variations in chromosomes, or sometimes we can call as chromosomal aberrations also. So the change may occur either in the structure of chromosome or sometimes number also. So number is also one of the change in the structure is also one of the parameter. The structural chromosome rearrangements are changes in the physical structure of the chromosome. That means before cell division process and after the cell division process, we can observe the physical structure of the chromosome. It leads to alteration in the sequence of the genes or order of the genes, or sometimes one chromosome lose its genes, another chromosome receive more number of genes. So the allelic variation we can easily observe due to these mutations in particular genes. So chromosomal aberrations are substantial changes in the chromosome structure. So these structural chromosome changes are divided into mainly 4 categories. So there are 2 primary ways in which the structure of chromosome can be altered. How? For example, in the diploid cell, the chromosomes are arranged in pair model or sets. If any haploid set of the chromosomes, after the change, one of the chromosome containing number of genes is more than previous. So that is nothing but changes are involved in the total number of genes in the chromosome. In another aspect, the changes are involved in the genetic gene composition, gene number is same, but the genes are arranged in different sequence or different order. That means one is change in the total number of genes, in another aspect rearrangement of the same number of genes. సో ఇక్కడ ఏం జరుగుతుందంటే క్రోమోజోమల్ విపతనాలలో క్రోమోజోమ్లో ఉండే జన్యువుల సంఖ్యలో మార్పులు జరగచ్చు, క్రోమోజోమ్లోని జన్యువులు అదే సంఖ్యలో ఉండి రీఅరేంజ్మెంట్ జరగచ్చు. సో ఏం జరిగింది అన్న దాన్ని బట్టి బేస్డ్ ఆన్ హౌ దీస్ క్రోమోజోమ్ స్ట్రక్చర్ అండర్గో చేంజెస్ we can broadly classified into four types. First one is deletion, second one is duplication, third one is translocation, fourth one is inversion. So first we can go for first aspect. The total amount of genetic information in the chromosome can change. That indicates what? Before and after. Before change and after the change, we can observe what is the physical change in the chromosome. So in the before chromosome is the homologous chromosomes contain 300 genes. After the change, a homologous chromosome contain less number of genes, another homologous chromosome containing more number of genes. So in that way, we can observe the change. So next slide, we can go for next slide. Okay. Based on these chromosomal aberrations are grouped into two major kinds, variation in structure and variation in number. Just we can say like that. In 1926, H. Stuttevant discovered the inversions in chromosomes, and also in 1923, C.B. Bridges discovered the duplications, deletions, and translocations in the chromosomes. So what is meant by deletion? First, we can go for what is meant by deletion. Deletion is nothing but we can call as deficiency also. Deficiency, when we can call as deficiency, when deletion of a small portion of a chromosome resulting in loss of one or sometimes more number of genes. So for example, just we can draw the diagram of a chromosome. So based on that, we can easily explain. So this is the centromere. So this is the two arms of the chromosome. So at the time of cell division, these two arms are divided into two chromatids. Yes, two chromatids like this. Okay. So in this chromosome, either chromatid part or sometimes chromosome, it lost a small portion of this part of chromatid. So it is nothing but loss of some genes from this chromosome. That is called as deletion. So when we can call deficiency, the terminal part of the chromatid is removed by any type of factor. So at that time we can call as deficiency. The intercalary part of the chromatid is missing. That is called as deletion. So the deficiency originates from breakage of occurring at random in both chromatids of a chromosome, called as chromosome break. If it is both the same chromatid, same portion is removed or by cut, it will be released from the mother chromosome. The terminal parts of the reunion takes place. So if the deletion occurs in one chromatid also sometimes, so the breakage may be caused by various agents like radiations, sometimes chemicals or drugs and viruses at the time of during the cell cycle. These deletions occur in maybe somatic cells or in germinal cells also. అంటే శారీరక కణాల్లో క్రోమోజోమ్లో డిలీషన్ జరగచ్చు లేదా బీజ కణాల్లో కూడా జరగచ్చు. So depending upon its location, a deletion may be terminal when a single break takes place. So ఇప్పుడు ఇక్కడ కట్ అయింది అనుకోండి, that is called it require only one break. One break is enough to remove the terminal part. But if the intercalary part of the chromosome, it requires how many breaks? Two breaks. Two breaks are needed to remove this intercalary part of the chromosome. So the interstitial part of the chromosome is also deleted by break at 2 points occur in middle portion of the chromosome. So this is called as deletion. So deletion is nothing but loss of genes either in single or in few number by deletion. These deletions are again classified into terminal deletion and intercalary deletion. So after deletion, how the chromosome behaves? So just you can observe in heterozygous condition, both terminal as well as intercalary deletions takes place, can be observed during the Pocketing stage of meiosis by formation of a buckle-like projection. For example, this chromosome, this is the one homologous chromosome. So it is not undergo deletion, but another homologous chrome deletion takes place. So definitely we can observe based on the size of the arm. So this part is very small length when compared to the another homologous chromosome. If intercalary part of the chromosome part deleted, in the corresponding homologous chromosome, this will be formed like a buckle-like portion. This is called as buckle-like portion. So this is called as loop. So loops are formed at the time of pocketing because bivalents are formed. In the bivalents, the corresponding homologous genes are missing here. So that's why in opposite homologous chromosome, a buckle-like projection is formed that indicates what the deletion takes place in one chromosome. So based on that, we can easily detect the chromosome whether it is undergo deletion or not. So these deleted part of the chromatid or chromosomes we can study it by some cytological methods or cytological studies also. So good examples for this deletion, just we can the effect of a deletion depends on what was deletion. So homozygous wild type organism may give a normal phenotype, while the same deletion in the wild type allele of heterozygote would produce a mutant phenotype. So deletion of the centromere resulting is an eccentric chromosome that is lost. Usually without centromere, the chromosome cannot survive, and it causes detrimental or very serious lethal consequences in the organism. So good example for this deletion, Cray-Duchart syndrome. This is a very one of the important point for the entrance point of view. Cray-Duchart syndrome. So Cray-Duchart syndrome observed in the human beings by the deletion in the short arm of the fifth chromosome. Short arm of the fifth chromosome. So when fifth chromosome, a small part of the chromosome deleted, the infants are cry like cat. So that's why this syndrome name is called as Cray-Duchart syndrome or Cray-Duchart syndrome. Another one is the 22nd chromosome deletion caused in the long arm. It causes Philadelphia chromosome, that is called as Philadelphia chromosome. Why it is called as like that? Due to the long arm of the 22nd chromosome, the intercalary part missing in that chromosome. That's why it is called as Philadelphia chromosome. So next one is duplication part. So what is meant by duplications? So duplication is nothing but a presence of same block of genes more than once in a haploid complement is known as duplication. So what is that meaning? That a sequence or a block of genes, which is repeated in haploid complement, that indicates what? So this is a chromosome which containing a series of genes. So for example, A, B, C, D. So in this haploid complement, a block of genes repeated more than once. That means ABC series of genes, again it is repeated either in the same, beside the same genes, or in a different position, in a different location also. That means ABC genes, sequence of ABC block genes repeated in the haploid chromosome. So that is called as duplication. అంటే ఒక నిర్దిష్ట వరుస జన్యువుల సముదాయము ఒక హాప్లాయిడ్ సెట్ క్రోమోజోమ్లో ఒకటి కంటే ఎక్కువ సార్లు పునరావృతం అవుతే దాన్ని మనం ఏమంటాం? డూప్లికేషన్స్ అంటాం. వీటిని ద్విగుణీకరణాలు అని చెప్పి మనం పిలుస్తాం. సో ద క్రోమోజోమల్ ఫ్రాగ్మెంట్, హౌ ఇట్ ఈస్ పాసిబుల్? in one chromosomal fragment is detached and it add to the homologous chromosome at one or at exactly in the terminal position, or maybe in the intercalar position it attached, then we can get more genes in that chromosome. అంటే ఇందులోంచి ఒక బ్లాక్ ఆఫ్ జీన్స్ కట్ అయినాయంటే దీంట్లో నంబర్ ఆఫ్ జీన్స్ తగ్గినాయి అన్నమాట. ఆ జీన్స్ దాని హోమోలోగస్, the corresponding homologous chromosomes attach అయినాయి కాబట్టి దీనిలో number of genes ఏమైతే పెరుగుతాయి. For example, before it is 30, both the homologous chromosomes contain 30 genes are there. But in this -3 is there, so it becomes 27 genes. It becomes 30 plus 3, 33 genes. That means the changes observed in the genetic information represented by the gene number only. So in this chromosome, the gene number is increased. In another haploid complement, the gene number is low comparatively. So whenever there is a duplication in a chromosome, there is a corresponding deletion in another chromosome. ఎక్కడైతే క్రోమోజోమ్లో డూప్లికేషన్ జరుగుతుందో దాని కరస్పాండింగ్ హాప్లాయిడ్ క్రోమోజోమ్ కాంప్లిమెంట్లో ఏం జరుగుతుంది? డిలీషన్ జరుగుతుంది అన్నమాట. అంటే డూప్లికేషన్ ఇండైరెక్ట్లీ కన్సిస్టింగ్ డిలీషన్ ఆల్సో. The origin of duplication can be traced to unequal crossing over during meiosis. ఇది ఎలా జరగచ్చు అంటే అన్ఈక్వల్ క్రాసింగ్ ఓవర్ వల్ల జరగడానికి స్కోప్ ఉంటుంది. అంటే జనరల్గా 2 నాన్ సిస్టర్ క్రోమాటిడ్స్ ఈక్వల్ లెంత్లో కట్ అయితే మనకి ఇక్కడ డూప్లికేషన్ జరిగినట్టు కనిపించదు. కానీ అన్ఈక్వల్గా, అంటే ఒక దాంట్లో లార్జ్ సెగ్మెంట్, రెండవ దాంట్లో షార్ట్ సెగ్మెంట్ కనుక ఎక్స్చేంజ్ జరిగితే దీనిలోని జీన్స్ దీనిలో రిపీట్ అవుతాయి అన్నమాట. సో దట్ ఈస్ నథింగ్ బట్ ఇట్ ఈస్ కాల్డ్ డూప్లికేషన్. The mechanism ensures after crossing over between non-sister chromatids, equal exchange products are formed. But if paired chromosomes are misaligned means అవి ఒకే వరుస సమాంతరంగా అమర్చబడకపోతే it is not possible for exchange to take place between exactly opposite locations on chromatids. అంటే వాటి అలైన్మెంట్ సరిగా లేనప్పుడు అన్ఈక్వల్ ఫ్రాగ్మెంట్స్ క్రాసింగ్ ఓవర్ జరగడానికి స్కోప్ ఉంటుంది. కాబట్టి ఇక్కడ డూప్లికేషన్స్కి అవకాశం ఉంటుంది. డూప్లికేషన్ డు నాట్ ప్రొడ్యూస్ ఎనీ డ్రాస్టిక్ కాన్సిక్వెన్సెస్ యాజ్ లైక్ యాజ్ deletion in term of phenotype and survival. The deletion sometimes it leads to lethal. That means it causes death of the organism also. So next duplications types. We can go for duplication types. So duplications are again classified into 3 types. One is tandem duplication, another one is reverse tandem duplication, third one is displaced duplication. So what is meant by tandem duplication? See here, a block of genes repeated more than once in the same order. In the same order. For example, see here, a block of genes is here. This D and E is gene here. Same sequence is repeated continuously. So that type of duplication is called as what? Tandem duplication. Tandem duplication. Tandem duplication is nothing but these two genes in the same order, it repeated more than once. That is called as tandem duplication. Reverse tandem means the order of the genes in the first D and E, whatever the deletion pores from the haploid set, it attached in the reverse position. That means we get ED, but D is twice, E gene is also twice in that chromosome. That is duplicated. But the whatever the portion of the chromosome attached to the haploid, opposite haploid complement in reverse order, that is called as reverse tandem. Okay? Displaced on the same arm. Displaced and in a different position. D is here. So D is towards left side of the centromere. The deleted part is also attached in the same arm, that is called displaced. Sometimes it is displaced on different arms also. So from the centromere, this is the centromere, left side DE genes are there, but these duplicated genes are here in the opposite arm, that is displaced. Sometimes it is attached to the non-homologous chromosome is also, that is called as displaced duplication also. So reverse tandem, tandem, displaced. These are the different type of duplications. Is it clear? So for this example is, good example for the duplication is the Drosophila eye-shaped allele bar gene that reduces the number of eye facets, giving the eye a slit-like rather than oval appearance. So this is the normal wild type of the Drosophila melanogaster. The bar gene represented in a 16A and 16th A region. So the eye shape is oval. When these two chromosomes undergo duplication, see here, the 16A part is duplicated in this chromosome. The shape of the eye, it look like a slit model. And double bar type, that means the 16A region of the chromosome is repeated in haploid complement thrice. 16A, 16A, 16A. So these three regions indicate the eye shape is small and it convert into slit-like model. So due to the duplication of the chromosomes. So this is the best example of Drosophila melanogaster, the bar character of the eye. The bar character is a dominant X-linked character. It controlled by 7 bands of 16A region of X chromosome. Yeah, next. So different kinds of duplication loops. So the opposite strand lost the genes. This way, that's corresponding area of the haploid complement changed into a buckle-like projection. That's why it is produced loops. So in the loop, BC genes are there. In the normal part, BC is there. So in this way, we can observe after the duplication, here BC part is duplicate, and it at the time of meiosis it forms loop-like structures. Next, third one is third type of chromosomal aberration, that is called as translocation. What is meant by translocation? So translocations are nothing but a kind of chromosomal rearrangement in which a block of genes from one linkage group is transferred to another linkage group. Another linkage group. That means I am already told at the time of linkage, in every organism, the haploid chromosome number is equivalent to the linkage groups. In these linkage groups, a block of genes are shifted from one linkage group to another linkage. See here, the integration of chromosome segment into a non-homologous chromosome is known as translocation. For example, X is equal to 4. That means 4 pairs of chromosomes are there. So a block of genes are deleted and it shifted to another chromosome, not the same haploid chromosome. That's why it's called non-homologous chromosome. It involves the transfer of a segment of chromosome to a different part of the same chromosome or different chromosome. So, for example, this color indicates one linkage group. This color indicates another linkage group. A block of genes cut and shifted to the or rearrangement. Here, the total number of genes are equal. There is no change in the haploid set of the chromosomes. So, for example, in the first two cases, one of the chromosome lost its genes. So this chromosome united with normal chromosome. In the progeny, some genes are lost. So, but here the total number of genes are same, but the sequence or arrangement of the genes are changed in this haploid sets of chromosomes. So that is, you can remember when compared to the deletion and duplication. So types of translocation: simple, another one is reciprocal, another one is shift. Simple is nothing but an attachment of a small terminal segment of a chromosome to the end of a homologous chromosome or to a non-homologous chromosomes. So, ante oka chromosome loni terminal part cut ayi non-homologous chromosome loni part loki attach aithe dan manam అని చెప్పి పిలుస్తాం. Reciprocal means chromosome and is followed by the requires at least 3 breaks in the chromosome. Among these, reciprocal and shift are the most common types. So we can explain with the help of diagrams. It is easily to explain these translocations. So this is the deletion. This is a chromosome containing A to F genes. So when it is undergo inversion, this type of resulting seen in the chromosome. When it is undergo duplication, we can see this type of result in the progeny, and deletion and insertion. So here translocation is C here. So this is one homologous chromosome. This is another non-homologous chromosome. Here A, B, C, but DEF is missing. So the DEF is shifted and OPQ part is shifted to non-homologous chromosome. So just based on that, we can easily explain about the 3 type of translocations. Simple translocation, in each case, a small segment of chromosome is added to the end of the other non-homologous. అంటే ఇక్కడ మనం సింపుల్ ని ఎట్లా చెప్పొచ్చు అంటే, this is one chromosome. Okay, this is another chromosome. We can go for with a different color. So it cut here the terminal part and it is attached here, and both the haploid side, that means total number of genes in the linkage group may not change, but arrangement of the genes are changed. This is called as simple. Shift translocation means it requires broken off as is inserted within the break in another non-homologous chromosome. That involves 3 breaks, 2 in one chromosome, 1 in non-homologous chromosome. In case of reciprocal, vice versa. It is exchange of parts between 2 non-homologous chromosomes, as for example, exchange of segments between 2nd and 3rd chromosomes of Drosophila. Good example. So this is the 3 types of translocations are generally observed in the Drosophila. Okay. Next, we can go for 4th type of chromosomal aberration. Inversion, that is nothing. Inversion is nothing but the chromosome segment rotate with 180 degrees from the original orientation. So 180 degrees it will be rotate, and breaks takes place where it is break at the exact in that place reunion takes place. So based on the inversion, based on the inverted segment of the chromosome, which is having centromere or not, based on that, this inversion types again divided into 2 types: pericentric inversion, paracentric inversion. Pericentric means the inverted chromosome segment, this is the chromosome actually. So this chromosome bends like this. This is the 180 degrees. Okay. So in this place, it will be break. And exactly in this same position, it will be reunion takes place. So that's why the gene order in this fashion, it will becomes like this. So here A, B, C, D is there. And here is cut. So E, F, G, E, F, G, H. So after AB, we are getting here G, H like that. So because that will be rotate with how many degrees? 180 degrees from the original orientation. So if the inverted segment containing here centromere, we can call as pericentric. The without in the not in it is present in the inverted segment, it is extremely end, that is called as paracentric. So pericentric, paracentric inversions are leads to changes in the centromere positions. So a metacentric chromosome undergo inversion, it converted into submetacentric chromosome. So like that, we can go through the diagram also. So linked genes are often inverted together. The meiotic consequence depends on whether the inversion occurs in homozygote or heterozygote. A homozygote will have normal meiosis, but when a heterozygote undergo this inversion chromosomal aberration, at the time of crossing over, it produces different type of gametes. With normal gene sequence and inverted chromosome sequence, inversion loop formed during the meiosis, it causes different class of gametes also. Inversion loop in heterozygote forms tightest possible alignment of homologous regions. So this is the electron microscope photograph indicates the chromosome undergo inversion process. So gametes produced from pericentric and paracentric inversions. See this, these 4 type of gametes are produced due to the inversion process: normal product, deletion product, and deleted product, inversion product. So different type of class of gametes are formed due to the crossing over takes place, and also inverted segment is involved because every chromosome having 2 chromatids. So one of the chromatid DNA segment undergo the inversion, and the remaining part is in the normal way. So in this way, we are getting normal product as well as the duplication of A arm, deletion of D arm. Like this, these photographs are indicates different class of gametes are produced. So already we learned about the translocations also. Okay. So this is about the structural variations in the chromosomes. Apart that, we can also discuss about the numerical aberrations or numerical changes in the chromosome also. So generally, every plant species or animal species having a unique chromosome number. It may not change. Universally, every organism containing a unique number of chromosomes. But sometimes at the time of meiosis, due to the abnormal behavior of the chromosomes or any failure in the movement of the chromosome with the help of spindle fibers, or at the time of ATP is not available to uh, pull the chromosomes towards the polar region. Some physiological and biological factors influence the abnormal behavior of the chromosomes. The abnormal behavior of the chromosomes leads to change in the number of chromosomes. So based on that, we can these numerical changes are classified into euploidy and aneuploidy. Aneuploidy means the basic set of chromosomes either add or loss of chromosomes 1 or 2 or sometimes 3. Euploidy means in any organism, the sets of chromosomes are increased. For example, after meiosis, we are getting only one single chromosomes. It may not pair with the corresponding homologous. That means haploid, monoploidy. All sporophytes are diploids. But in the crop plants, example, banana is a triploid plant.